Okay. Very good evening, boys and girls. Hope you all are doing well. So let's get connected today again with another session of MCQ with us. So today I'm going to take MCQs on nerve muscle physiology. Right? So today we'll be having few discussion on the questions based on nerve muscle physiology. Right? So I'm your mentor at Unacademy, Dr. Reena Kaur. Yes, you are mentor at Unacademy, Dr. Reena Kaur. So let's get started with the first question. Yes, in a normal, healthy muscle, what is going to occur as a result of propagation of an action potential to the terminal membrane of a motor neuron, right? So your options are opening of the voltage gated calcium channels in the presynaptic membrane, depolarization of the T tubule always will result in muscular contraction, will going to cause increase in the intracellular calcium concentration in the motor neuron terminal, or E, that all of them are correct. So come up with your answers, boys and girls. A, B, C, D, E. Yes? Yes. So the correct answer is definitely E, that this is the sequence what occurs, and you know that we have studied this as the excitation contraction coupling, right? We have studied this as the excitation contraction coupling, and remember the coupling agent is calcium. So the neuromuscular junction is equipped with a so-called safety factor that ensures that every impulse that is traveling from the terminal of the motor neuron is going to result in action potential in the sarcolemma. So when a healthy muscle is, contraction is ensured. The voltage sensitivity of the calcium channels in the presynaptic membrane and the high concentration of the extracellular calcium ensures that there's going to be sufficient calcium which will occur, and it will come inside, which will cause the synaptic vesicles to fuse with the presynaptic membrane and release in the acetylcholine, and this acetylcholine is going to guarantee the depolarization of the postsynaptic membrane and end in an action potential. So if you remember that this is our neuromuscular junction, right? This neuromuscular junction, yes, this is the thrown into folds, and this is the axon or the action potential traveling, and here is going to be the voltage gated calcium channels. So this voltage gated calcium channels will open and calcium will come inside, and this calcium is then going to result in the fusion of the synaptic vesicles with release of acetylcholine. Remember that this has got various proteins, that is the syntaxin and the V snare proteins. These proteins are interrupted by the botulinum toxin, which prevents the release of acetylcholine and resulting in the flaccid paralysis, right? And eventually these acetylcholine is going to bind on these receptors, resulting in the end plate potential, which will then develop an action potential. This action potential will travel along the sarcolemma and it will dip into the T tubules. These T tubules has got DHP, that is dihydropyridine receptors, which in the case of skeletal muscle behave as voltage sensors. Once these open, they're going to cause a conformational change in these L-tubule rhinodine receptors, and there is going to be release of abundant calcium, which is then going to bind on the troponin C. The troponin tropomyosin complex will be shifted, and you're going to get the actin myosin cross bridge. So this entire process is what we called as the excitation contraction coupling, and the coupling agent is calcium, to be remembered, right? Moving next. Now, a 65-year-old man sees a neurologist because he has got weakness in the leg muscles. that improves over the course of the day or it improves with exercise. So this is a very important clue being given to us that the person is having muscle weakness, but this muscle weakness is improving with exercise. When you take extracellular electrical recordings from the single skeletal muscle fiber, it shows us a miniature end plate potential. Low frequency stimulation of the motor neurons, however, elicites an abnormally small depolarization of the muscle fibers. The amplitude of the depolarization increases after exercise, right? So remember that this is a case history being given to us, and now we're going to look into the question behind it. So the amplitude of depolarization is increased with exercise. This is a very, very important clue. Now the question says that based on these findings, what is the most likely cause of the patient's leg weakness? A. Acetylcholinesterase deficiency, blockade of postsynaptic acetylcholine receptors, impaired presynaptic voltage sensitive calcium influx, inhibition of calcium reuptake into sarcoplasmic reticulum, or reduced acetylcholine synthesis. Yes, so the very important clue being given is that exercising is improving the symptoms, right? So it is of two things. You have to remember whether rest is improving the symptoms or whether exercise is improving. So this becomes C. That means there is impaired presynaptic voltage sensitive calcium channels, and you remember that if it is E, that means if there is something which is causing improvement with the work done, it takes us towards Lambert-Eaton syndrome. If it is a Lambert-Eaton syndrome, it is going to be present presynaptically. That means it is going to affect the voltage gated calcium channels. So remember the miniature end plate potential. What is miniature end plate potential? It is insufficient, right, packaging of acetylcholine. The most likely explanation in this case is definitely a presynaptic deficiency, impairment of the voltage sensitive calcium channels. So because there is lack of the voltage gated, accumulation of calcium, that is going to decrease the trigger of release of acetylcholine. Right, now the question have given us a clue that increase in the postsynaptic depolarization is observed after exercise. This indicates that with repeated stimulation there is accumulation of calcium, and when there is accumulation of calcium, it is going to bring about release of sufficient amount of acetylcholine, and the acetylcholine then can bring about depolarization. So remember, if the condition says that there is improvement with rest, right? If there is improvement with rest, the clinical condition goes more towards, or the picture goes more towards myasthenia gravis. But if it tells us that there is improvement with work, or there is improvement with exercise, or there is improvement with repeated stimulation, then this takes us more towards Lambert-Eaton syndrome. Right? So an important thing how we can categorize and differentiate. Next, muscles that are best suited for high intensity exercise but brief. Okay, brief high intensity exercise contains which type of fibers? Mostly glycolytic, mostly slow twitch oxidative, that is red, or a mix of slow twitch red and a fast twitch red, or a mix of glycolytic white and fast twitch. So remember, the type of exercise being performed is short duration but high intensity. Short duration but high intensity. So the correct answer is going to be D. Now, the mixture of fibers that is ideal for this type of exercise, that is a short duration and high intensity, is going to be definitely a mixture. See, this is what is the size principle, which helps you to understand the recruitment of motor units, right? Recruitment of motor units. That means when the person is suppose standing, or he starts walking, or he starts running, how is going to be the recruitment? how is going to be the recruitment. Yes, how is there going to be the recruitment? Recruitment is going to be from the slow fibers, right? From the slow fibers. Yes, the recruitment is going to be from the slow fibers, then type 1A followed by type 2A followed by type 2B. That means if you have remembered that we have studied the types of muscle fibers, right? The type of skeletal muscle fibers, we've classified them into type 1 and type 2A and 2B. So when we are performing and when the intensity of the work increases, there is going to be recruitment of slow, that is type 1 fibers, followed by type 2A and then type 2B. So type 2A basically are FOG, that is they are fast oxidative glycolytic, whereas these are FG, that is fast glycolytic, whereas slow fibers are basically the oxidative fibers. So this is how there is recruitment of the motor units with increase in the intensity of work. Now, a 32-year-old woman undergoes appendectomy and develops malignant hypothermia following halothane anesthesia. So the clue over here is halothane anesthesia. What changes in the skeletal muscle causes body temperature to increase in this condition? So there is an increase in the body temperature. The options are an increase in the refractory period of alpha motor neurons, inability of the skeletal muscle cells to repolarize, excess calcium release from the sarcoplasmic reticulum during muscle contraction, production of endogenous muscle pyrogens, or repeated firing of the presynaptic terminals of alpha motor neurons. So what is the clue given to us? That is, there is malignant hyperthermia and that is induced by halothane anesthesia, and therefore the correct answer becomes C. So you know that with malignant hyperthermia, which is a life-threatening increase in the metabolic rate and body temperature, is attributed to the mutation of the ryanodine receptors in the skeletal muscle. These ryanodine receptors which are present in the L tubules. Okay, so the ryanodine receptor or the calcium release channel on the sarcoplasmic reticulum opens when the skeletal muscle is activated, and we know that yes, Darshika, correct, the correct answer is C. The flow of the calcium through this is actually responsible to bind to troponin and initiate the muscle contraction. Now because there is mutation, the calcium channels will open. There's going to be abundance or a continuous release of calcium ions. This is going to result in increase in the metabolic activity, and there is going to be increase in the body temperature. So because of this mutation of the ryanodine channel, there is an uncontrolled release of calcium. So this is something important. So you can just see over here that these are the L tubules and the T tubules. In this T tubules, we have got the DHP receptors, but here in the L tubules, which are the sarcoplasmic reticulum cisterns, you have got the ryanodine receptors. Right, you have got the ryanodine receptors, and malignant hyperthermia is a mutation of this channel. Next is a 26-year-old medical student walks briskly down the hallway to grand rounds while he is eating a hamburger and fries on the way. What is the major difference in the contractile response occurring in the smooth muscle and the skeletal muscle? So the question is depicting you two things. One, that the person is eating. He is eating means there is going to be some kind of motility in the GI tract. Second is he is walking. That means there is also involvement of the skeletal muscle contraction. So now you have to tell me that what is the difference in the contractile response of the smooth and the skeletal muscles. The mechanism of force generation, the nature of the contractile proteins, the role of calcium in initiating contraction, the source of activator calcium, or the source of energy used during contraction. So the major difference is C, that is the role of calcium in initiating contraction. That is a biggest difference in the skeletal muscle and in the smooth muscle. So the difference is the excitation contraction coupling that is occurring. You remember that in the skeletal muscles, right? In the skeletal muscles, the calcium is going to bind to troponin C, and this is going to uncover the active sites of actin. Okay, it is going to uncover the active sites of actin, and this is then going to form cross bridge with myosin. But if you remember the smooth muscles, the troponin C is absent in the smooth muscles, and what is present is calmodulin. So there is going to be a calcium calmodulin complex which will activate the myosin light chain kinase, leading to its phosphorylation, and then myosin cross bridges are going to be formed with actin. So the major difference in in the contraction of skeletal muscles, or you can say in the contraction of the striated muscles and the non-striated muscles, is in the striated muscles the calcium is going to bind with troponin C, whereas in the non-striated the calcium is going to bind with calmodulin, and therefore the whole mechanism is going to change. So this calcium calmodulin is associated with myosin. Therefore, it is called as a myosin-linked contraction or a myosin-linked activity in non-striated. And in the striated, this troponin C is bounded to actin. Okay, the troponin C is bounded to actin, and therefore it is called as an actin-linked contraction, right? An actin-linked contraction in the striated muscles. Yes, moving next. Which of the following activities depicts an isometric contraction? You know what is isometric contraction? The tension is increasing but the length is remaining constant. So which is this type of contraction? Lowering different amounts of weight, right? Passive stretching like you do in warm-ups, running downhill and then uphill, squeezing the gluteal muscles together, or walking down a flight of stairs. Yes, anybody would like to answer? A, B, C, D, E. Which one of them is typically an example of isometric, that is tension increasing but length remaining constant? Yes, so the correct answer is D. When you are squeezing the gluteal muscles, you are not doing any work, you are just increasing the tension of the muscles. Whereas lowering the weights, stretching, running, walking, these are all examples of isotonic exercises where the tension is remaining constant and the length of the muscle is decreasing. The length of the muscle is decreasing because there is shortening of the sarcomere, and as the individual sarcomere shortens, there is going to be shortening of the muscle itself. So the muscles need to increase tension without movement, and this is done when you are holding the buttock muscles tightly. Remember that this is especially what we called as the length tension relationship. It is telling you about the active tension and the passive tension. The points over here to be remembered. So active tension is mainly because of the contraction, that means actually because of the actin myosin cross bridge activity, right? Whereas passive tension is basically stretching of the series elastic elements and the parallel elastic elements of the muscles. That is the stretching of the tendons, the stretching of the connective tissue, and very important, tighten, that is elastic protein of the muscles. Next. Yes. Now, this is an example of a sequence based question. Boys and girls, you get this type of question in exam. It is a typical example of a sequence based question. Excitation contraction coupling is a process where the muscle is stimulated to contract, right? And then there's going to be muscle relaxation with the following steps being given: 1, 2, 3, 4, 5. You have to tell me what is the correct sequence of this activity. So options: action potential, calcium release, cross bridge cycle, cytoplasmic calcium removal, and depolarization of the T tubules. So what is the correct sequence? Yes, the correct sequence is B. What is B? 1. That is, first there is going to be action potential, followed by 5. This action potential is going to result in depolarization of the T tubule, followed by 2. That means calcium released in the sarcoplasm, followed by 3. Once the calcium is released, there is the cross bridge activity, which is going to result in the muscle contraction, and finally there is going to be removal of calcium, that is 4, which is going to result in the muscle relaxation. So boys and girls, this is a very, very typical sequence based question and a recent pattern MCQ, which also comes in the AIMS pattern of exams. So remember, wherever there are sequences, okay, wherever there are events, they can come as a sequence based question. Like one of the example is at NMJ. Now, which of the following changes to calcium handling would prevent cardiac muscle tissue from contracting? Right? So you have to tell me that what is going to prevent the cardiac muscle tissue from contracting. Increasing the extracellular calcium concentration, increasing the opening time of dihydropyridine receptors, increasing the opening of ryanodine receptors, inhibiting the calcium sodium exchanger, or sequester all the extracellular calcium. So anybody would like to make a try? A, B, C, D, E. Yes. So the correct answer is E. That means when you sequester all the calcium, there is going to be muscle relaxation, right? There is going to be muscle relaxation. Now, in the cardiac muscle, the calcium influx from the extracellular space is brought about by dihydropyridine receptor, and in the skeletal muscles, in the skeletal muscles, the DHP receptors mainly behave as the voltage sensors, right? They mainly behave as the voltage sensors, whereas in the calcium, calcium induced calcium release, is causing muscle contraction, especially in the cardiac muscle. So this is the very, very important difference, that in the cardiac muscle, the T tubules are not only behaving as voltage sensors, but they are giving an important source of ECF calcium. This ECF calcium results in getting more calcium from the L tubules, that is from the ryanodine receptors. So this is called as the calcium induced calcium release in the cardiac muscles. Therefore, when you sequester all the calcium back, the contraction will not occur. And again, remember that in the cardiac muscles, the relaxation is brought about by the circa pump, that is the calcium ATPase pump, right? There is also presence of a sodium calcium exchanger, and apart from this, there is also present of phospholambin. This phospholambin, when phosphorylated, increases the calcium or takes the calcium back into the sarcoplasmic reticulum and ceases of the contraction. So these three things are responsible for the muscle relaxation as far as the cardiac muscles are concerned. Another important thing to be remembered, when you want contraction to occur, there should be increase of calcium in the sarcoplasm, or that is the cytosol, plus ATP, right? Plus ATP. And when you want that there should be relaxation, you require ATP, but you have to decrease the calcium in the cytosol, right? You have to decrease the calcium in the cytosol, or you can say that you have to increase the calcium in the sarcoplasmic reticulum. That means you have to pump the calcium back into the sarcoplasmic reticulum with the help of ATP. So this have come many a times as an MCQ, and sometimes it becomes very confusing. So remember, sarcoplasm, that is cytosol, where you are increasing the calcium and increasing the contraction. But if you want relaxation, definitely you require ATP, but you have to decrease the calcium in the cytosol, which is occurring by increasing the calcium in the sarcoplasmic reticulum. Right? And also in the cardiac muscles, you have got SERCA, pump sodium calcium exchanger, and phospholambin, which are responsible for the muscle relaxation. Phospholambin is present only in the cardiac muscles. It has got not been presence in any other muscle tissue. So it is important as far as the cardiac muscles are concerned. Now, a muscle biopsy is taken from a healthy volunteer, which is showing large amount of myoglobin rich glycogen poor fibers with many mitochondria. So there is myoglobin rich glycogen poor. That means there is basically presence of type 1 muscle fibers. So which of this could be? It could be paraspinal, biceps, deltoid, or pectoralis major. Yes, the correct answer is A, that is paraspinal. Remember, the type of muscle fibers we are speaking is type 1, which are red oxidative fibers, which are present in the postural muscles, right? Because they are fatigue resistant. Fatigue resistance. Therefore, the correct answer becomes paraspinals. Yes, correct, Rita. It is paraspinal. Dhaniksha, biceps से भी ज़्यादा better answer would be paraspinal because they are kind of postural muscles. Got it? Dakshika. The posture maintaining muscles like the paraspinals have got high content of type 1 fibers, which have got numerous mitochondria, myoglobin, high capillary density, and function mainly by oxidative, that is aerobic metabolism. The other options, rapid forceful arm movements, which will have more percentage of type 2 muscles as compared to type 1. And initially also, in one of my other classes I have told you that any muscle, if you consider, is not made up of any single type of muscle fiber. A muscle fiber is a mixture of red and white fibers depending upon what is the activity performed by that muscle. Yes, a 62-year-old man presents with hypokinetic movement disorder, which is characterized by paucity, characterized by slowness of the voluntary movement. That means there is a kind of bradykinesia. Which of the following characterizes the order of recruitment of muscles during normal muscular movement? Same, we have studied what is called as the size principle, and the question is asked in a different way. So the options are: anaerobic fibers are recruited before aerobic, fast muscle fibers are recruited before slow, large muscle fibers are recruited before small, poorly perfused fibers are recruited before richly perfused fibers, or weak muscle fibers are recruited before strong muscle fibers. That means every time you must remember that the classification of type 1 and type 2 muscle fibers should be very nicely known to me. It should be very nicely known to me, right? So therefore the correct answer becomes E, that the weak muscle fibers, the type 1, the slow, the oxidative, they are first recruited, followed by the type 2, which are fast, glycolytic, anaerobic, having larger diameter, large conduction velocity, a fast twitch, action potential is fast, they have got a slow, fast twitch, that means the muscle contraction relaxation is fast, whereas the type 1 has got a slow twitch. right? So please, again I repeat that this is a very high yielding topic and the topic which comes very times repeatedly. So please nicely study the difference between the type 1 and the type 2 muscle fibers. Yes. So small, weak, slow, fatigue resistant muscle fibers which are innervated by the small spinal motor neurons, whereas the large spinal motor neurons, they innervate the large, fast, strong and easily fatigable muscle fibers, that is type 2. The slow fatigue resistant, they have got dense capillary network for perfusion, lots of mitochondria and oxidative metabolism is their choice. Next. A 56-year-old woman presents with a flat red rash on the face. Okay, a flat red rash on the face and on the upper trunk with an erythematous rash on the knuckles and in the shape of a V on the neck. She complains of muscle weakness, especially when in conditions when she's pulling a chair or when she's combing her hair. Laboratory findings show increased creatine phosphokinase, positive anti-nuclear antibody, anti-Mi2 dermatomyositis specific antibodies. In addition to oral prednisolone, physical therapy is ordered with the goal of improving the muscle strength and thus ability to perform her daily activities. So the entire question is just a question to make it lengthy, right, to make it more confusing and to give a clinical scenario, a typical question of INICT pattern. And if you look into the last line, how does repetitive stimulation of the skeletal muscle fiber causes an increase in the contractile strength is the physiology which is asked behind. So a very typical question which is asked, that a clinical picture being given, right? So the correct answer is definitely B. That means when you are increasing repetitive stimulation of the skeletal muscle fibers, you are increasing the duration of the cross bridge cycling. Remember, more the cross bridge cycling, more is the strength, more is the active tension which is developing. So each time the muscle is stimulated by an alpha motor neuron, enough calcium will be released from the sarcoplasmic reticulum to activate all the troponin within the muscle. Every cross bridge is going to result in increase in the muscle tension. So repetitive firing, increase amount of sarcoplasmic reticulum stretching, maintaining the cross bridge cycling, that means increasing the calcium within the myoplasm. Number of myofibrils activated are more, right? So, and because of all the cross bridges activated during each time the skeletal muscle is activated, an increase in the calcium concentration would have no effect, right? So the important thing to be remembered over this is the clinical history with a simple physiology being asked. Next is a 16-year-old adolescent boy on the track team asked his pediatrician that if he can take creatine on a regular basis in order to increase the muscle strength prior to a track meet. Which of the following explains that he would prefer to take creatine? That means what is the normal function of creatine? Creatine delays the metabolism of fatty acids. Creatinine increases muscle glycogen concentration. Creatine increases plasma glucose concentration. Creatine is converted to phosphoryl creatine, or creatine prevents dehydration. Yes, so the correct answer is D, that is creatine is converted to phosphoryl creatine. Now this phosphoryl creatine will be converted to ATP. So when the metabolic demands exceed the rate at which ATP can be generated by anaerobic metabolism of glycolysis, phosphocreatine will supply the necessary ATP. An increase in the concentration of the phosphorylcreatinine in the muscle increases the amount of ATP and therefore increases the performance, right? So muscle energetics, types of muscle fibers, right, and energy sources, these are also high yield areas, right? These are again high yield areas. So boys and girls, with this we conclude today's session. So we'll be meeting again with more and more MCQs. Till then, happy learning. Please always remember to rejoice so that you are always restarting, refocusing towards your goals with enough refreshment. So be persistent, and thank you all. Our toppers, special class features, we are starting up with our batches, our various subscriptions being allowed. So thank you.